Hello dear students, welcome to the botany online class. Today's topic is endosperm development. So what do you mean by endosperm and how it will be formed within the seed? So endosperm, it is a nutritive tissue which is formed within the seed after fertilization. So during reproduction of the flowering plants, what happens is the anther means it is a part of a stamen, right? So the anther produces the pollen grains, and this pollen grain carries two male gametes. Am I clear? Whereas the female sex organ means the pistil portion of the flower contains the female gamete. Okay, so during pollination, what happens is the anthers are the pollen grains are released from the anther and are deposited onto the stigmatic surface of the pistil. So during germination, this pollen grain produces the pollen tube, and the pollen tube, it will enters into the style region, and finally it will reaches the embryo sac. Okay, and this pollen tube carries two male gametes. After reaching the embryo sac, the male gametes are discharged within the embryo sac, and of these two male gametes, one of the male gamete fuses with the female gamete, nothing but the egg cell, to form a diploid structure called zygote. Whereas the another male gamete, it fuses with the secondary nucleus to form a triploid structure called primary endosperm nucleus. Okay. Now this zygote, it will develops into an embryo, whereas the primary endosperm nucleus will develops into endospermic tissue. Am I clear? So endosperm, it is a nutritive tissue found in the seeds of nearly all the flowering plants after fertilization. When the second male gamete fuses with the secondary nucleus, it forms the primary endosperm nucleus, which is triploid. Now this primary endosperm nucleus undergoes repeated mitotic divisions to form the nutritive tissue called endosperm. Clear? So in this picture, we can clearly observe that the pollen grains during pollination are transferred to the stigmatic surface of the pistil, where this pollen grain on germination produces the pollen tube, and the pollen tube carries the male gametes. So of the two male gametes, one of the male gamete fuses with the egg cell to form the zygote, whereas the another male gamete fuses with the secondary nucleus to form the primary endosperm nucleus. Now this primary endosperm nucleus later on forms the endosperm. Clear? In this way, endosperm tissue is formed within the seed. Am I clear? Coming to the types of endosperm formation. Endosperm formation is of three types. They are nuclear endosperm, cellular endosperm, and halobial endosperm. So nuclear endosperm, it is also called as free nuclear endosperm. Means in this type, the first division and usually several of the following divisions are unaccompanied by the wall formation. Okay, that means here, now let us consider, this is the primary endosperm nucleus or primary endosperm cell, and this primary endosperm cell, it contains a single nucleus. During a nuclear type of endosperm formation, what happens is the nucleus of the primary endosperm nucleus or primary endosperm cell, it divides repeatedly to form many nuclei. So here only the nuclear divisions takes place, not the cytoplasmic division. Am I clear? So after repeated divisions within the primary endosperm cell, it produces many nuclei. Later on, a central vacuole develops so that all the nuclei are migrated to the peripheral position. Am I clear? So this type of endosperm formation is called as nuclear endosperm. Here only nuclear division takes place. Clear? The nuclei may either remain free or in later stages they may become separated by walls. And examples for nuclear endosperm formation: wheat and coconut. Coconut water. So coconut water is an best example for nuclear endosperm. Am I clear? So this is the picture of nuclear endosperm formation. Here you can see the first stage. In the first stage, this is the primary endosperm cell or primary endosperm nucleus, which is formed by the fusion of secondary nucleus and male gamete. So after the formation of primary endosperm nucleus, the nucleus, it divides so that it produces many nuclei. You can see here only the nuclear divisions takes place, not the cytoplasmic division. Now, after the formation of many nuclei, central vacuole develops so that all the nuclei are shifted to the peripheral position. Clear? So this is about nuclear endosperm formation. Coming to the second type, cellular endosperm formation. So in this type, what happens is there is cytokinesis after each nuclear division of the endosperm nucleus. That means, so this is the primary endosperm nucleus with its nucleus. So this primary endosperm nucleus, it undergoes nuclear division so that it produces two nuclei. Soon after the division of nuclei, so here wall formation takes place. Okay, so there is cytokinesis after each nuclear division. Okay, so again the nuclei divides, again the wall formation takes place. So in this way, the nuclear division is followed by the cytokinesis. Okay, and the endosperm thus has a cellular form from the very beginning because first and subsequent divisions are all accompanied by cell wall formation. Example for cellular endosperm formation: petunia and coconut. Coconut meat or solid part or the kernel of the coconut is an example for cellular endosperm, whereas liquid part of the coconut is an example for nuclear endosperm, and the kernel, the white kernel, it is an example for cellular endosperm. Clear? So you can see here, immediately after the divisions of nuclei, the wall formation takes place so that the cell becomes cellular. Okay, it's a multicellular structure. Moving on to the third type of endosperm formation, that is halobial. It is an intermediate type between the nuclear as well as cellular types. So here the first division is accompanied by cytokinesis, but the subsequent divisions are free nuclear. The chamber towards the micropylar end of the embryo sac is usually much larger than the chamber towards the chalazal end. So halobial means it is the intermediate type between nuclear and cellular. So this is the primary endosperm nucleus. So during the first step, what happens is the nucleus of the primary endosperm cell, it divides to form two nuclei. Immediately wall formation takes place here. So now this is a type of cellular endosperm. Okay, the single celled primary endosperm nucleus becomes two cell structure here. Okay, now the smaller cell is the chalazal cell. Okay, whereas the bigger cell is located towards the micropylar end. So the bigger cell is micropylar cell and the smaller cell is chalazal cell. Later on, the nucleus in the chalazal cell as well as the micropylar cell, it divides repeatedly to form many nuclei, but there is no wall formation. Understood? That means the type of endosperm formation is here free nuclear. Okay, once again I'll repeat. During halobial type of endosperm formation, the nucleus of the primary endosperm cell, it divides to form two nuclei. Immediately after the formation of two nuclei, cell wall formation takes place. So that now this is the type of cellular endosperm. After a cellular type of endosperm formation, the nuclei which are present within the chalazal cell as well as the micropylar cell, it divides repeatedly to form many nuclei, but here the nuclei remains free. Okay, and example for this halobial type of endosperm formation, Hydrilla, Vallisneria and order Halobialis. So these are about the types of endosperm: nuclear, cellular and halobial. Am I clear? Coming to the last topic. So this is the picture of a halobial endosperm formation. You can see here the formation of chalazal cell and the micropylar cell. Okay. The last topic is functions of endosperm. Endosperm is important for the growth of an embryo and stores the reserve food. So this endosperm performs the function of supplying nutrition to the developing embryo. And the endosperm contains cytokinins. It's a type of plant growth hormone. Cytokinin means it's a type of plant growth hormone which regulates cellular differentiation. For the process of differentiation, cell differentiation, cytokinin is important. So endosperm, it's a nutritive tissue. It supplies nutrition to the developing embryo and it stores reserve food materials and also contains cytokinin which regulates the cellular differentiation. Okay, so these are about endosperm development. Thank you.